Okay, let's get started. Good evening, everyone. Thank you all for coming today. A lot of people today. And I'm Abhishek. I'm a postdoctoral fellow in Mriganka Source Lab. And it's a real honor for me to be able to introduce Professor David Hubel and Professor Wiesel here at MIT. We are truly delighted to have both of them with us here today, I believe after many years. So way back in 1959, working together in Steve Kuffler's lab at Johns Hopkins, they made what they later called an accidental discovery. And they found that the image of a glass slide, the edge of a glass slide, actually made cells fire in a cat's striate cortex. So this simple observation actually led to their first paper in the good old British Journal of Physiology, and which was followed again by a continuous flow of papers, outstanding papers, which really defined the field of experience-dependent cortical plasticity. Professor Hubel and Wiesel provided a quantum step in our understanding of how visual system works, along with Roger Sperry of Caltech, as you all know, that they won the Nobel Prize in 1981 for their discoveries concerning information processing in the visual system. In 1978, they were awarded Louisa Gross Horwitz Prize from Columbia University. So very briefly, introduction would be, Professor Hubel studied mathematics and physics as an undergrad at McGill, and then entered medical school there. And then he moved to US to work with, initially at the Walter Reed Hospital in Washington, DC, and then joined Steve Kuffler's lab. In 1959, he moved to Harvard along with Steve and Torsten and stayed there ever since. He became the chairman of the department in 1967, where he's currently the John Franklin Enders Professor of Neurobiology Emeritus at Harvard Medical School. He's still actively doing research and teaching. Professor Wiesel, on the other hand, came from the other side of the pond. He began his scientific career in Karolinska Institute, Sweden, where he received his medical degree in 1954. And in 1955, he moved to US to work with Stephen Kuffler. By 1973, he succeeded Kuffler as chair of the Department of Neurobiology, the newly formed department. And in 1983, he joined the faculty of Rockefeller University as the head of the Laboratory of Neurobiology. He was named the Rockefeller President in 1991 and the President of IBRO in 1998. And since 2000, he has served as the Secretary General of HFSP, Human Frontier Science Program. He won also numerous awards, and most importantly, National Medal of Science in 2005 and Ramon y Cajal Gold Medal in 2006. So we are also very fortunate to have our very own Professor Mark Beer, who will be moderating the discussion. So Mark's own work has shown that the synapses in the cerebral cortex are modified by experience and how they store information. So it's great to have him. Mark has been a Max Planck Fellow initially, won an Alfred Sloan Award. He's been an HHMI investigator and an elected fellow in the American Academy of Arts and Sciences. So please join me in giving a warm welcome to all three gentlemen.
Thank you, Abhi, very much for organizing this entire event. I had nothing to do with it, so we really all appreciate your effort on this. And I just want to make a few brief remarks because really I turn it over to Torsten and David. But I thought I would give a brief personal story of how their work influenced my scientific life and my career. They published their first paper together in 1959, which was 2 years after I was born. And it took me a few years to catch up on the literature, but eventually I did. And I started reading their papers in the 70s when I was an undergraduate at Duke University. And they immediately became my scientific heroes. And why? Well, they revealed the beauty of the cerebral cortex and how structure and function were related. They dared to simplify, and they wrote their papers in a way that I could understand. they made it clear that there were still big discoveries to be made and really encouraged me to go into this field. So I went off to graduate school, and then I was seduced by the visual cortical plasticity paradigm, which is actually schematically shown on this slide. So they had shown that manipulating the quality of sensory experience in a young kitten could dramatically modify the organization of the cortex. And so I and really a flood of my contemporaries went into this field of visual cortical plasticity in the 70s and the 80s. And there were so many big questions left to address, such as, which synapses are modified by deprivation? How do synapses lose strength as a consequence of deprivation? What's the biological basis of binocular competition? What's the basis by which there's a compensatory strengthening of the connections from a non-deprived eye? And what's the basis of the critical period? Why does this plasticity fade with increasing age? So although I never trained with David or Torsten, I do feel that I'm a scientific descendant and one of their children. I'm not sure they would own me, but that's what I'm claiming. As I said, there was a whole wave of people that entered the field because of their work, and particularly in this area of visual cortical plasticity, which included Carla Schatz, who I see here in the audience, and Mrganka Sur, Larry Katz, Mike Stryker, and a whole bunch of us were in the field to pursue these very interesting questions. And like a good son, occasionally I would look to Torsten or David for some sign of approval of the work that I was doing. But I must say that the children did have a tendency to squabble quite a bit. And I think that like wise parents, they stayed above the fray and didn't pass judgment on any of us. And I think we're going to work it out in the end, so don't despair. So to prepare for this, I looked at their Nobel lectures last night, which are available online. And if you haven't seen them, you should. They're really inspiring. But in the introduction to Hubel's lecture, they were likened to two hemispheres of the same brain, describing their 25 years of great work together in collaboration. And David corrected the introducer by saying, no, they're more like two oxen pulling the same cart. But to me, they're examples of great partnerships, famous partnerships, such as Hodgkin and Huxley, and Watson and Crick, and even John Lennon and Paul McCartney. So, as I mentioned, Abi was responsible for this event, and he really had to twist my arm to participate in this role. Because, it's like being a little boy wearing a Superman t-shirt and then being asked to introduce Superman to a group of peers. So I thought that to give you some small tastes, some small appreciation for what it's like to be in my role today, I just play one little quick video clip and then we'll turn it over.
Man, everyone knows who you are. You're Paul McCartney. Oh, it's great to be here. You remember when you were with the Beatles? Sure. That was awesome. Yeah, it was.
Okay. So you guys remember when you worked together? It was awesome. And so now, actually, I'd like to turn it over. I think, Torsten, you're going to get teed up first to make a few remarks about the early days.
Thank you for turning off the lights. It's good to be here. I should to comment what you just said. I think that, initially we were approached by Abne, who introduced us first, to have an informal discussion with students about the early days. And so we thought that could be nice. And we have, even if there are more people now than we had expected or anticipated, we are still intent to have an informal discussion, sort of, about the early days. And we're going to show some pictures, we may make some recollections, but we are not going to be serious. I mean, for those of you who know the work, for those who have never read our papers, or it might be difficult to enjoy it. So you heard already, I think, as David suggested, we both should say a few. You know a little bit about our background, but it might be useful to have some personal notes before we came together. What happened to us before we came together? So maybe, David, you could start with your...
Yeah. Yes, when Steve, I was at Johns Hopkins for a while as a resident in neurology, and the Hopkins had a wonderful thing called the doctor's dining room, where everybody had lunch there, and all the people interested in neurology or neurosurgery or neurophysiology would tend to sit at the same table. And that I guess was where I got to know Steve and various other high priests in the field, like Vernon Mountcastle and so on. Then I went to Walter Reed, as Torsen has said, and had 3 years there. It was a very small group of neurophysiologists and neuroanatomists, and it's really where I got started. I think when I first went there, they expected me to do EEG, which I had had some training in Montreal, and I had ended up making a solemn vow that I would never again look at another EEG. So I told them this at Walter Reed, and they sort of smiled and said, that's fine, what would you like to do? And of course, I had no idea. I had never done any research at all. If you can believe that, even in summers. So I was sort of cast adrift and could do whatever I wanted. And I played with devising a new electrode that turned out to work well. And I think I fished around and a number of...
This is a famous tungsten electrode that had been used all over the world since David designed it.
Yeah, I never made any money on the electrode.
Too bad, yeah.
Anyhow, I recorded from a number of things: spinal cord, dorsal cochlear nucleus, auditory cortex, and I think in the end I decided the visual cortex would be at least, if not more fun, the easiest, because it's right underneath the skull and easy to find. And so I got nowhere at first, and in fact, I discovered that cortical cells visual cortical cells don't respond to visual stimuli, because I made the same mistake as other people had made before by shining a bright light into the animal's eyes and seeing no effect at all on the cells. And one day, in a fit of desperation, these were chronically implanted cats I was working with. They were awake behaving, and I stood there and went like this and found that the cell fired like a machine gun when I went in one direction and not at all in the other. So I was onto something, but of course, being basically a stupid person, I never tried this. Anyhow, I got out of, when my time came close to the end at Walter Reed, Steve Kufler...
there was one other thing that David discovered in these early days, and that he recorded also from the auditory cortex.
Yes.
And he noticed that he found that attention, he crackled the...
Yes. If I held up my hands with 2 pieces of paper and rattled one of them, the cat would look at that, obviously taking an interest, and every time I rattled it, the cell would respond. This was in the auditory cortex, and not respond to the other piece of paper in the other hand. And so I didn't really know whether this was attention or not. The head of the lab wrote a paper called Attention Units in the Cortex, which made me rather angry because I didn't think I had established anything. For all I knew, the animal was pricking up his ears and having them face the stimulated paper. This has never been settled. Anyhow, Steve Klooffer wrote me a letter and said that would I be interested in coming to Hopkins, back to Hopkins. And I had, the plan was that I should go into, join Mountcastle's department and set up a visual research there. But Mountcastle was...
Maybe you can say a few words about Vernon.
Yeah, well,
many of the young people may not...
already said that he was a high priest in the field, and probably, to my mind, one of the most impressive people in the whole of neurophysiology. But anyhow, I was supposed to go to that lab and set up my own lab there, but they were remodeling his lab, and so Steve said that he had a postdoc, Torsen, whose colleague had left to go to the West Coast, and would I be interested during that period of remodeling in joining Torsen and working with him. And of course this was terrific, and this was a world center of retinal physiology about which I knew very little, maybe not exactly nothing. But anyhow, I went there and we started to work together. I remember going home the first night and telling Ruth, my wife, that I thought this collaboration was really going to work. It was very clear that Torsen had an attribute that I lacked, and that is he is very fond of hard work, and he kept on. Every day we would start out and Torsen would say, "What should we do?" And of course I had no idea what we should do. And anyhow,
I'd like to insert here that my first, David had already presented some of the work he did on the cat, moving his hand, and there was a presentation. And those days, in the old days, the meetings were in Atlantic City. That disappeared by then. But, so, and we all went. And it was before the Society for Neuroscience existed. So, and so I went to the session and listened to David. And I realized that this was a person of obvious interest. And then later on, my colleague I worked with on the retina before that, we visited David in Walter Reed, and he showed us how to make tungsten electrodes. And so that was our earlier contact before we then met. And actually, when Steve wrote to David and asked him if he was interested in joining, then we had a meeting, Steve Kuffler, David, and I, to discuss what we were going to do. And that was the time when we actually decided that we knew something about the retina from Kuffler's work and from I had been working on the retina with a colleague, the other postdoc who left, by the name of Ken Brown. And so we said, and I did some single cell recording in the ganglion cells. So we decided it would be fun to see what happened if you record more centrally from the lateral geniculate body, the relay nucleus, and the cortex, because nobody knew anything about it. It was unexplored territory. The reason why it was unexplored was that just a few years before, people have made it possible to record from single cells in the brain. for technical reasons. First, David's tungsten microelectrode was a very important step in this development. And then also, there were other people who developed micropipettes, and this kind of electrode also started to be used. But we were very fortunate. We came into the field at the stage when it became possible to actually study single nerve cells in the cortex and in other parts of the brain. And part of it is technical reasons that maybe David, he developed a chamber, and maybe the background to that is also interesting, I think, because if you open the skull, the animal is breathing, and there's a lot of pulsations, because suddenly, in the brain you have a certain pressure to stabilize things. So you open it up and it does this. So there was a scientist at Hopkins by the name of Davis who developed and showed that you can if you have a chamber on the cortex, you can stabilize it. So David then developed a very elegant method which now is used all over the world also, to record from single cell first in anesthetized animals, but the basic technique is now used in all the experiments using awake animals, where you have a chamber and so on. But that principle, I think David was the first really it developed when he was with Jasper in Montreal. He didn't mention that, but
Right.
But that was part of it. So I just like to point to my colleagues the fact that he developed not only electrode, but also the closed chamber, that then also is a very important tool for anybody interested in studying single cells in the cortex.
Yeah. I think one of the first days that I went to work in Steve Kufler's lab, maybe I should say, I walked in the first day to Steve's office, and typical Steve, he said, "Welcome, David. Take off your hat. Take off your coat. Take off your hat. Do up your fly." And he had this business of being able to make a joke out of almost anything out of midair. Anyhow, the first week I was there, Torsen had done some work a lot of work recording from retinal ganglion cells, and in fact from every kind of cell in the retina with Ken Brown. Steve had a paper in the mid-50s to the effect that the center surround organization of retinal ganglion cells changed if the animal was dark adapted. That is, if the only rods were at work rather than both rods and cones. And the take-home message of that paper was that in dark adaptation, where you only were dealing with rods, the surround dropped out and you only had a center. And I think this is probably what most people still think. What Torsen did, taking me aside that first week, was to explain that he had made it pretty clear that this result was wrong. This was a paper by Barlow, Fitzhugh, and Kufler, the famous Barlow, the great-grandson of Darwin, and a big contributor to the field. And in fact, it is wrong. If you're slightly above threshold, even though you're way in the dark adapted state, it's perfectly clear that you get a weaker response to a big spot than to a small. And so it's curious that has persisted, probably because the major name of the authors of this paper, I think the only paper I know of where Steve was wrong isn't quite the right word, because you could read the paper many times and not make out quite what they were saying. But anyhow, Steve worked in every area, and he was, in a sense, the head of the lab that Torsen and I were in, but it was a different organization entirely from a typical lab today. where Torsen and I were completely independent. And Steve, of course, was independent, worked with one colleague or two. He gave me some papers to read the first weeks I was there, 3 papers on presynaptic inhibition, I think it was. And I read the papers over a whole weekend and made many comments. And he completely revised the papers. And of course, it wasn't even thought of that my name should be on that paper or that any of us should be have our names on any other papers. So Steve, all those years, though his input was wonderful in criticizing our papers, as Torsen will point out in a few minutes, but it didn't enter. It would have been ludicrous to think that his name should be on our papers. This has completely changed, of course, today, and I can say more about what I think of present-day organization of labs, in case you don't know.
Maybe before that, I would like to, I mean, we are here at MIT, and when it was interesting, when David and I, when we did our original work, recording from cells in the cortex, at the same time here at MIT was this gentleman. Yeah. And it was interesting because his way of trying to understand, there was a paper he wrote for the frog's eye tells the brain that if you're a student here, you should look at, because it's a classical paper because it shows the imagination, the clarity of mind, and the imagination of designing the experiment, which is classic. So he died a few years ago, but he was a legend here and still is at MIT. So I just wanted, sort of, because when we came to Boston, to Harvard, people assumed that we knew each other and we knew about each other's work, but we didn't, actually. And but it was kind of nice, and we became good friends and had many discussions. He did what the frog tells, the eye tells the brain, with Maturana, who was a Chilean scientist, who then went back to Chile. But so that was part of the reason why I wanted to show.
Interesting to think back that when we first came to Harvard, Harvard compared with Hopkins seemed completely stuffy as a place to be, and maybe it was the medical school and the propinquity of the Peter Brent or Brigham Hospital, where everybody was, they acted like high priests. Anyhow, the one thing that Torsen and I both found was the welcoming that we got from MIT, and particularly the psychology department at MIT, led by Luke Teuber. Harvard psychology department was of no use to us at all, because it was besmitten with operant conditioning, and the high priest there, am I surprised?
Oh, he was. I like Skinner, but
yeah.
But there was the whole strong emphasis on psychophysics and behavior and type of psychology. Not so much interested. Teuber was very interested in biological mechanism explaining behavior. So he was an early cognitive. He wasn't recognized at those days as a cognitive scientist, but he came out of the medical field. So and his interest in behavior was based on his experience with neurological patients, but he became a very extremely good leader here at MIT of the department, and is really the founder of the current department in brain science here at MIT.
Yeah, he made it the leading psychology department in the world, I think, probably. Yeah. Maybe we were biased because we had no use for operant conditioning and all that sort of stuff. But the atmosphere in the lab led by Steve Kufler was terrific, and Kufler, I think, was suddenly invited to come to Harvard to take over the teaching at Harvard Medical School. And so he brought all of us with him. And there were, I think there's a slide showing.
Yeah, I have a few. I made a few slides. This is David at the time, roughly where we are talking about, and this is this was taken before I came to the United States, so I probably will be smiling. You see, and. This is a picture taken at Hopkins when we just started out doing our research. A kind of young people looking into the future, hoping, and full of energy. And this is the first experiments we did. And we found an old blackboard, put some sheets on, and we found an old slide projector, then we had an oscilloscope amplifier, but that was our equipment. And with that equipment, we made our main discovery about and I like to show this. No? I don't know how to press. Where is the computer?
Maybe I could just mention at this point that our slide projector was the successor of that one. It was a light slide projector.
So this is a geniculate cell. This is a cell that feeds into the brain, okay? You record from a single node cell, stimulate this light in a receptive field. And this is the center of the receptive field. You stimulate the surround, it is inhibited, give an off response. That was known beforehand for the retina, had the same kind of responses. So that was nothing new. But we need to know what fed into the cortex because before we could record from the cortex, obviously. What happened? So you take away surround, you take away inhibition that is caused by the laser. And this is an important for when we record from the cortex. This is just a slit of light that moves in different orientation, and the response is the same, independent of orientation. So this is now a cortical cell, and you can hear it a different pitch, and we map this receptive field of the cell, very simple, on the board. It's very crude, but that. It's very crude. We didn't get orientation quite right, and we didn't get precise. But
it took us many years before we even thought of drawing a graph of responses as a function of orientation of cells. We thought all this was foolishness.
So this was part of the Nobel Prize, right there. The fact that you can stimulate the same part of the retina in different orientation, and you have cells specified in the cortex too.
I was just going to say that we at one point. That's a dental spatula that happened to be sitting around.
It just shows how robust it is. Very, nothing. You don't need statistics to see it.
One day, Torsen and I decided we'd look at dark adaptation, and we had a 750-watt slide projector with a lot of light escaping from the bottom. And so to make the room completely dark, to get rid of this stray light, we went to some sort of store and bought a big sheet of black cotton and put that over the slide projector, except of course for the lens. And the chairman, we were at that point in the Department of Pharmacology at those first years at Harvard. And the chairman of the department was Otto Kreyer, a very kind man, but being German, he was rather serious. And we had this, we were recording then and made the room completely dark, and that all worked fine. And a knock came in the door, and Otto Kreyer came in just at the moment that the cotton burst into flame. that was exciting, and we, Torsen and I thought it was screamingly funny, but Otto Kreyer didn't share that feeling. But it's hard not to remember that. This is. Yeah.
So this is just to show Steve Kuffler and David. That was again at the time when this we wrote. Once we had made the discovery with orientation, we wrote an abstract to be presented at one at Atlantic City, probably. And we showed it to Steve Kuffler, and he made some correction. I don't know if he got the corrections in.
Yeah, they're there.
They are?
If it's possible to find something on this. I came in the next day. And
do we have
Torsen,
that's maybe it's among David's slides.
Torsen was sitting at his desk and he looked rather glum. And he said, "I don't think Steve thought much of our abstract." And here is a copy of the abstract. It's I hope it's one.
Yeah, no.
It's on that little disc. It's sort of worth waiting for. I show it to my students when I'm preaching about the importance of writing.
It's in David's slides. It's his slides, I think.
No, I'm afraid it's not there.
Where is it? The nice lady who helped? I don't know where.
It was on a separate little disc that I gave out, and I think.
Let's go back to the where we were. You can't have everything in life.
Can't have everything.
I hope this works because I spent half of yesterday taking a picture of Steve's comments on our abstract.
So anyhow, there was. We wanted to show this picture because I wanted to say for the young people here the importance of a mentor, because he left us alone, but because of his nature, his intelligence and his sharp sense of wit and understanding of issues in scientific as well as other issues, he became like a father figure to us, a mentor in a good sense. We trusted him. But he never interfered with what we did. And and I sort of always feel that it's so important to when you go, particularly as a graduate student, to find, and the postdoc, and to find the right environment, to be mentored right. And so we were fortunate in the funding and coming into a situation where this was possible. Now, we had moved to Harvard, and there was what we call the gang, and we were first in the Department of Pharmacology by this man Kreyer, and it soon became clear that the gang was, we gave a fantastic course in neuroscience, to neurobiology, to the medical students, and so on. So before too long, Steve Kuffler convinced the dean that we needed to create a department in neurobiology. And I think that was one of the first departments in the country in neurobiology. And that department then became very famous. That was in 1968, 69 that was created, and it still exists. And we have Some of us here have been, Eric Fischbach is here, he was the chair also, like I was for a while, and Carla Schatz was also chair. But these people, this is, these two guys, you know, this is Ed Kravitz, he's sitting in the audience, and then it's Ed Forsman and Dave Potter, both for those in neuroscience. And it was we wanted, again, to be productive and creative. I think to have an environment that is supportive, but also a sense of fun. Because science should have the spirit of fun and games to some extent. And I think we were fortunate. And we have spent the day today at McGovern Institute, and I got a sense again here that, you know, a spirit of coming together because we all care about science and enjoy it is important.
It's interesting that
Here it is.
Yeah.
Very good, thank you, Mark.
That's
David, you make the comments.
Yeah, well, Torsen looked a bit glum in his sentence, as I said. He said, I don't think Steve thought much of our abstract. and so, needless to say, when the abstract finally appeared in publication, it had no resemblance to the original. And it was very much more. I haven't gone through this word for word. It would be fun to actually have our abstract alone and Steve's corrections alone, but I use this as a...
And the final copy, yes.
Yeah, right. When I'm preaching about writing, I usually show students this because it's important when you write something to show it to other people and to a wide variety of other people. That's really how you learn to write. And I guess all of us in this group that you've seen, when we wrote papers, would hand it around to other people. And I remember I had one result, and in the margin, Ed Firstman wrote bullshit. So this is the nice thing about teaching in those very earliest days at Harvard were that there was area teaching where students did nothing but 6 weeks of neuroanatomy and neurophysiology. And there were other areas like guts and glands and things like that. And it was certainly, I think, the most popular course in the medical school. And I think it was so popular that the rest of the faculty, in their wisdom, faculties like to change the organization of teaching, as you probably all know. And so this lasted 4 years or something like that, and then the faculty got rid of it and went to a Monday, Wednesday,
It wasn't
Friday.
the faculty, it was the dean.
Was it the dean?
It was the faculty who didn't tend not to make such decisions.
Yeah. Anyhow,
I like to. You may get the impression here that the initial writing was very bad, but and it was clearly not perfect according to Steve Kufler. But in my book, David in our collaboration, David has been the writer. I mean, he has an exquisite style, in my view, of writing and clarity and so on. And so when Mark said that when he could read the paper because he could understand what was being said, I think the credit to large extent should go to David.
But it has to be said, I tended to write the first draft, but I've never seen anybody who could hit the nail on the head in criticizing what you've written. And it was typical he would take a sentence and move it earlier in the paragraph or later or something like that. But it was always a terrific improvement. So if you wanted to get the best criticism around, Torsen was the guy to show a paper to. But it's one thing I've been interested in lately is comparing the organization of our little department with present-day organization. And I keep telling myself how happy I am that I didn't start out now doing neurophysiology, because there's this tendency when you're the senior person in a group, you find yourself no longer doing research and instead sitting at a desk raising money. And this is certainly the case in our department and probably typical of departments all over, at least in this country. So this is something I've preached about and become unpopular in our department because people don't like it to be said that they're no longer doing research. But every group wrote their own papers, and there was no longer a system. In order to have your name on a paper, you actually had to twist the dials in a group, and there wasn't this tendency that the last author on the paper may not have done any of the experiments but raised the money. So I find this unfortunate, but anyhow, this is
another picture of just to give you a sense of the collegiality and how much we enjoyed each other, company and discussion. This is Eric Frank, somebody may have heard of. He's now at Tufts, and Eric Schwartz, he's at Chicago, and Stewart is at Duke. At the Charlottesville. Yeah, USC, that's right. This is, I like this picture because it's sort of, we spend the summer in Woods Hole, David and I don't know, David was probably out. I don't think David in the picture, but we spent summer at the Sol, and the people and we went to the beach, and this picture was taken. There's David Van Essen to the left, and then Seymour Benzer. Many of you probably knew Seymour, who was a wonderful person who just died a few years ago. And this is a German postdoc with Steve, and then Steve Kufler's electronic guy, and then me and Steve. And I like the picture because it, again, brings out this whole idea that we were colleagues, but we also enjoyed life in different ways. So this is just the last picture I think I have, similar to the one that you saw before, when we received at the press conference, and I don't think I have any. Yes, this picture is. I visited. I live in New York and Sweden, and David outside Boston, Newton at the time, and David is an interesting man in many ways, but he plays piano. He's quite a good piano player. He plays a flute, but he also likes to weave, and. He is also very good at the lathes. All the machinery that he built for the electrode holders and all that, he machined himself very, very elegantly. So again, I think this kind of skill, inventiveness, and playfulness, I think is a very important characteristic, and I always admire this. And his weaving isn't bad, actually. So I can't turn this off. But I think for us the time that we never really saw each other socially. When our experiment, we started often early in the morning, 8 in the morning, and we got the animal, a cat or a monkey, and then prepared it from the beginning, anesthetized and prepared it, and then worked sometimes 24 hours or longer. And then at the end we perfused the animal. This was like a cute experiment in those days. And then we washed all the dishes and everything ourselves. And we kept on doing it. All the years we worked together, we actually did the experiment ourselves. And I think that's sort of, it's part because we enjoyed being part of the whole process, not telling you do this and you do that, but actually do the experiments ourselves. That's why we went into science, because we enjoyed David to make the machinery and to prepare the animal, all these features that it was part. So and the whole attitude I think we had was of exploring some, as I said before, when we started out, the microelectrode, the chambers, and all the possibilities to study cells in a living animal, single cells, made it possible for us to explore the unknown world. And so we talked about ourselves, we had no hypothesis. we just wanted to find out, what are the characteristics of these cells in the brain, and it was fun. I mean, it was actually, even it was hard work, it was a joy. And most of the ideas doing the long experiments, we talked to each other. Except very late at night when I was tired, I tend to speak Swedish, but David is very tolerant, so he.
Yeah, that's right. Yeah. But certainly, one famous experiment we did, famous in our minds anyhow, we started to record in a monkey, and there was a certain orientation of the cells, and we advanced the electrode, the orientation changed by about 10 degrees. And by the time we finished that penetration, we had gone around the clock twice in steps of 10 degrees. And when we finished, we realized that we had sat there without moving from our seats for 5 hours. And could hardly believe that it had gone on this long. That was, I think, the first time that we ever saw this precise organization in the cortex and into what turns out to be orientation columns. But the reason we had gone on for some years without any, sometimes hints that this was the case, but without any convincing evidence for it. And the reason was that we had selected our electrodes so that we could record nice big potentials from cells. And in order to do that, you have to have a selective electrode, and you advance for a while and see nothing, and then you see a nice big cell. And so the fact that we were choosing at random, as it were, cells gave us no idea of this organization. So when during that 5-hour experiment, we would have considered to be a lousy electrode at first, but we had units at every point in the penetration. That had allowed us to see that there was this fantastic organization in the cortex.
I think that Mark has been very patient with us. He's the MC, so now I'm going to, because some of you may now from this loose discussion if you want to have any questions to us, you should raise them.
I think you're doing a good job, Torsten, in running the show. But please, this is a conversation with the audience. So we have some microphones. We can run them around. So put your hand up, and I'm sure they'll be happy to answer your question. We can see what we can do. See what we can do. So while the microphones are making their way around, David, I have to ask you to comment on two things that I always remember about you or what I've read about you. One is the use of laboratory computers. And the use of laboratory computers.
That's David's specialty.
Yes. Yes, I remember the first computer that we got, it was a, do you remember the name of it?
It was 12, PD.
It was a great big
What was
thing.
it called earlier ones, do you remember? PDP 12, yeah.
Anyhow.
It was a big thing, console.
I remember thinking that I should get sort of on top of this and figure out how this worked. So I wrote a program where in which if I put in a number, it would give me back the number +1, and that took a whole day. And so I thought, if this is as far as I'm going to get with computers, we better as well give up the whole thing. And so, but in the end, it was deceptive, because we found the computers had one wonderful use, and that was that the heat control, air conditioning in the lab was very faulty, and we would sit shivering all day. And when it was really cold, we would simply turn on the computer. So, and I guess we made a sort of a thing of having a certain disparagement of statistics. We thought that if an experiment was so bad that you needed statistics, there was something wrong with the experiment. And I think this is probably largely true. I still don't know how to find a standard deviation or anything like that. But so
You have to remember that he was trained in mathematics initially. No, do you have a second question?
Yeah, the second concerns your advice to students who want to keep up with the literature.
yeah, my technique of keeping up with the literature, there was something, a publication that, what was it? It summarized, pardon? I can't.
Current content.
Anyhow, it was
Current content.
current contents, and Torsen subscribed to current contests, and he would mark the papers that he thought were worth reading, and I would get reprints of those papers, which in fact I would never read. It's not a praiseworthy attribute of me, but I really don't enjoy reading the literature at all. I think most papers are written in a way to make them maximally more boring, and what they never tell you is why the people are doing the experiment, how they got started. Any juice is removed from papers, and they end up dry as bones. So I don't read any more than I can help, but help did come, not from the Lord, but from Torsen.
I did
mark the papers that he thought
I did
were
my
worth
best to
reading.
keep. Yeah.
Yeah, I remember I read, I think you wrote this, that you went to Steve Kufler for advice on how to keep up with literature.
Yes, I complained how that I felt guilty about not reading more. Steve said you have to decide whether you want to be a producer or a consumer. And that was Steve's advice on keeping up with the literature. Maybe you're thinking of something else.
No, that's
but
exactly it.
I do remember that. Yeah.
Any questions? Come on, don't be shy. Here, come on. Can we use a microphone?
There's a microphone here.
Because we're going to record that. Don't mess it up.
You better be careful.
How has the pace of visual neuroscience research since your work, how has that matched up with your expectations? Did you expect us to be much further along, just where we are, or behind? So has the field progressed at a pace that you expected it to progress at the time you were doing your work?
I think it's, yes. Vision has become enormously popular as a field. I don't know whether that's because of our work. You could just as well say that it was Steve Kufler's or a number of other people. So we shouldn't take credit for that. What has always surprised me is that how lagging in central nervous system work the auditory has been as opposed to the visual, because the auditory system should be easily as interesting as vision. But if you look at books like Baer's famous textbook or Candel's shorts and so on, what's known about the central nervous system pathway of audition is practically nil, whereas you have chapter after chapter on the visual system.
I probably disagree a little bit. I think I mean, my own kind of feeling is that I'd hoped that the field would have moved forward more, advanced more than has been the case. And I mean, David and I, we set out to try to understand circuitry. I mean, we recorded from the nucleus, from the cortex, and the different layers, and we tried to inject cell and study the projections and connections in the brain as a model for how to study the higher functions in the brain. And so then people made very interesting discoveries, like, for example, phase cells in the infratemporal cortex. And this is very interesting, beautiful work, and important. But we don't know the circuitry. And this was now 30, 40 years ago, since no one has really gone to the trouble to really try to do, maybe people have tried but not succeeded, but I haven't seen any clear indication of a way of looking at the input and how it's being processed. Now we know there are 3 different phase areas, some relatively simple properties and some can generalize. You can recognize a phase in different orientations and so on. So this is a fascinating area, but one would like to understand, I mean, as a scientist, we want to understand how it's built, the mechanism behind it. And I think there's been a certain amount of failure there. Even depth cells, binocular depth cells, we know now that this is regionally separated out, but we still have rather unsatisfactory insight in the wiring. Color is still an area I think we need to learn much more about, even if David and Margaret Livingston, you did some work on the primary cortex, but and in the secondary V2 there are some, we still don't know a lot of work. Zeki did some very interesting observation using land stimulation, if some of you may remember, and that has never been really pinned down properly. So there are a lot of in vision, there are a lot of work to do for those of you who still have the energy and are willing to do hard work. And the interesting thing with the I think with the infratemporal lobe, the discoveries that Charlie Gross made initially was done on anesthetized animals, and so with such you could actually do circuitry work if you wanted to. So it's a lot of fun things to do. But I agree also with David that auditory cortex is amazingly that we know so little about. And if I was when I was on the picture looking forward into the future, that's probably would be a thing that one would try to do.
Part of the trouble may be the myth that in order to do science, you must have a hypothesis and proving it. Anybody who thinks that Galileo, when he looked at Jupiter and saw the moons, had a hypothesis, I think it's completely nonsense. So we've at least thought of our science as akin to geographical exploration rather than maybe, I don't know what scientists are to blame for that way of doing it. And of course, the hypotheses it isn't nonsense. But I think a lot of science isn't necessarily done that way. And when the study sections write as their critique of your grant proposal that it's not hypothesis driven, I think this is complete drivel as far as you do experiments to see what the result is going to be, and you don't necessarily have some high-flown hypothesis in the back of your mind. Although some science is done that way, it shouldn't be so one-sided that you don't get a grant if you don't have a hypothesis.
We do like to quote you on that, but I'm not sure it's been that successful.
I'm going to ask a question for a lot of the young people in the audience, and if you want to actually correct me, do it. But what do you think you would do if you were young now in this situation where to do any experiments you have to get through this incredible business, first of getting a job, but then of getting through a study section? I mean,
And you have to be 43 years old before you get to grant.
right. I mean, I'd like to think, I would like to hear what you reflect
A
on.
quick answer might be that I think I would go back to Canada. or Sweden or France. Part of the blame is the NIH. I think the NIH, had it not existed, we wouldn't have gotten going, because it was hard to write a grant that was so bad that it wouldn't be funded. But now, study sections seem to look for excuses to deny grants and breathe a sigh of relief when they found some reason to fail the grant request. And so
you have to remember, David, it's our colleague who make the reviews. So it's not a bureaucrat, it's actually the whole system, I think, has failed in many ways.
But David, your original research was funded by ONR, am I right?
That's right.
Not
Some
by
are.
NIH.
yeah, mine was funded partly by the ARC. I remember they had a man named Saveley, who was in charge of research for the Air Force, phoned me up and said, would you like, we can give you a grant of $10,000, which was a real fortune. And I said that I thought I had gotten a grant from the NIH, and so I said, I really don't think I'll need the money. And he said, can't you use some gravy? And so for the next decade or so, I was partly funded by the Air Force. I don't think that would happen today, but to have the financing so pivotal and terribly important and so time-wasting. I go around the department and all the leaders of groups are sitting in their offices. And they all sort of sigh and say, I wish I could get into the lab. But you feel like telling these people that if they're so that sad, they should go into the lab.
It's something, I mean, if you go around the country, many laboratories, you're head of a lab and there are 16 or so postdocs in the lab, and they all go out and they want to have grants and start their own laboratory. And I don't know if you read, Bruce Alberts wrote a very good editorial in the Science last fall, I think it was, pointing out that this system is not sustainable.
Yeah.
You can't keep on producing hundreds and hundreds of scientists like an exponential, and the funding stays like this. So the crisis we are in now is serious, and I think the only way to do it is to train fewer people. David and I, we trained 6 people in 7 people perhaps in 40 years. I mean, because we wanted to be sure that the ones we trained were good, and that they also could. And as David Potter, who is one of, he said, you don't want to reproduce yourself too often, which I think is a nice comment.
Torsen always had a good excuse for not taking somebody who wanted to come on as a postdoc or a graduate student. He would say, I don't think we have anything to tell these people. We don't know anything. So why would they want to come to our lab? I heard that over and over again.
So I have a question that I wondered if you think that we have earned now this bottle of wine.
I think that is the perfect ending to a fantastic afternoon. So thank you very much.
Thank you. Thank you.
Very nice.